Welcome everyone to the first lecture of MCB 182, which will be on generally a review on genes and genomes. So just as a reminder, please do not distribute the course materials online or anywhere else without my permission. And so the basic learning objectives for today will basically be to just kind of put everyone on the same page in terms of general background about transcriptional regulation, DNA, and genomics. Hopefully everything in this lecture will be a review for most of you. We still need to go through it because a number of you come from kind of non-traditional biology backgrounds, and so I just want to make sure we all are on the same page. And so the main topics that we'll be covering today will basically be things like why do we care about sequencing the genome, what is the genome, what's the basic structure of a gene, including intron, exon structures, and how splicing works at a pretty high level. I'm going to touch on basic kinds of regulatory elements like enhancers, promoters, repressors, and talk a little bit about where they're found relative to the transcription start site, as this will become more important as we talk about applications of genome sequencing to regulatory genomics. And I'm going to talk a little bit about knowing and kind of establishing which parts of the genome are so-called functional. And so the term functional is a little bit overloaded and somewhat controversial in the field of genomics. So I'll touch a little bit about that, and we'll discuss more about that later in the course. And we'll talk specifically about repetitive sequences and how even simple repetitive sequences can even themselves become functional. So it wouldn't really make sense to teach a class on genomics without briefly defining what the genome is. And so, of course, the genome in the context of this class is an organism's basically complete set of DNA. And so for humans, for example, generally speaking, excluding the mitochondrial genome, we have 23 pairs of chromosomes, and 22 of which are autosomes, and one pair of which are, of course, the sex chromosomes for most human individuals. And the human genome in general is approximately 3 billion nucleotides in length. And it's worth, to put 3 billion in perspective. Of course, most bacterial genomes have on the order of a few million nucleotides. And viral genomes, for example, can be much smaller than that. And so it's worth pointing out that genomes can vary pretty widely across different organisms. And so, for example, if you look at like a very macroscopic level and compare eukaryotic versus prokaryotic genomes, they differ pretty widely in terms of size. And so, of course, eukaryotic genomes tend to be much bigger than prokaryotic genomes. In eukaryotes, a lot of the DNA is non-coding. And so, for example, in the human genome, something like 98% of the genome is non-coding. And there tends to be a lot of structures like telomeres or centromeres in them. Whereas prokaryotic genomes are pretty compact in the sense that they're pretty gene dense. There's no centromeres. They don't have, for example, a lot of long-range enhancers. In terms of the chromatin structure even, eukaryotic genomes tend to be a little bit more complicated in the sense that the genome is typically wrapped around histones, and the positioning of these histones and their relative occupancy of different parts of the genome can actually act in a regulatory fashion. And so later on in the epigenomics lecture, we'll talk about how histone modifications and histone positioning can affect gene regulation. Whereas in prokaryotic genomes, there are no histones. And so, again, prokaryotic genomes, more, generally speaking, are much more simple in terms of their organization and gene regulation. And so also, as you probably know, eukaryotic genes oftentimes have introns, especially in the higher organisms like humans or other mammals. And even in terms of the ordering of genes, gene order tends to be conserved across species. And it's most importantly for this class, genes are monocistronic. And so what that means is that in terms of transcription, genes are typically
transcribed individually. And that's in contrast to in prokaryotic genomes, where genes generally don't have any introns, and oftentimes multiple consecutive genes can get transcribed together. And so, in other words, genes are often organized in polycistronic operons. As I mentioned on the previous slide, eukaryotic genes have promoters and also distal long-range enhancers that can complicate gene regulation, whereas in prokaryotic genomes, most of the gene regulation happens in the promoter region. And finally, in eukaryotic genomes, generally there's quite a bit of repetitive DNA sequence. And so, for example, in the human genome, roughly half of the genome is thought to have some kind of repetitive origin to it. Whereas in the prokaryotic genome, this is generally not the case. And so even in terms of how the cell is organized and how and where transcription happens, again, obviously in eukaryotes, transcription happens mostly within the nucleus, whereas translation happens outside in the cytoplasm. And in prokaryotic cells, this is not the case. There's no separate nuclear membrane and compartment. Something else that's pertinent to this course is the fact that genomes vary pretty widely in size and content. And so obviously on one hand, we have, for example, like the human genome, it's roughly 3.2 gig bases long, has quite a few genes, roughly, depending on how you count it, 20,000 protein coding genes, for example. Whereas the yeast genome in comparison is pretty compact at only 12.1 megabases. But, surprisingly, quite a few genes nonetheless, so somewhere on the order of 6,300 ORFs. It's also worth pointing out that genomes can vary pretty widely in their nucleotide composition. And so what that means is that some genomes, globally speaking, have higher GC content than others. And even within a given genome, like within the human genome, for example, the GC content of different regions of each chromosome can differ very widely. And so certain regions can be, fairly GC rich versus GC poor. And the reason that this is relevant is that GC content can have a pretty wide-ranging effect on things like gene regulation. And even for tasks like DNA sequencing, it turns out that the GC content of sequences can really affect how accurately that particular sequence can actually be sequenced. Right? And so really hot sequences that are very high in GC content tend to be harder to sequence using technologies like Illumina sequencing by synthesis that we'll talk about in the next lecture. And so as I mentioned on the previous slide, GC content can vary pretty widely across different genomes. And so here's just a phylogenetic tree with different organisms indicating what the relative percent GC content of different genomes are. And so the variation itself may not look that big in the sense that you can see that some organisms like the shrew are at 53.4%, and that's in comparison to, say, like orangutans that are at just a little under 46%. But you have to remember that given the size of these genomes, a difference of like 7% in GC content is actually pretty big. And so, of course, as you know, genomes can also vary in terms of their ploidy. And so obviously humans are diploid, whereas some plants can have much higher ploidy numbers. And the reason this is, again, relevant for doing genomics is that obviously when genomes have lower ploidy, that basically means that it's easier to call sequence variants, which we'll talk about in the next lecture. And it's also easier in terms of genome assembly. And so we'll also talk about assembly in the next lecture. But basically, one of the big tasks of genomics is to obviously sequence genomes. And so basically, organisms with higher ploidy are more difficult to sequence and assemble.
So in the context of this class, in terms of what genomics is, genomics will really refer to understanding the sequence, structure, and function of genomes, right? And so in terms of sequence, obviously, we need to be concerned about, what kinds of tools and technologies are typically used to determine the sequence of, the genome for a given organism, or at least a certain subset of cells within that organism. In terms of, the structure and function of the genome, we're obviously concerned about, mapping and identifying what different parts of the genome do. And so, for example, obviously that involves asking basic questions like, where are all, the set of all genes in the genome? How are they transcribed, and so on and so on. We're all also concerned about, what is the actual 3D configuration of the genome in the cell. And so in this class, we'll talk a bit about technologies to measure sort of structural properties of the genome. But we'll spend the large majority of the time asking questions related to the function of different parts of the genome. And so obviously, for example, protein coding genes, aren't the only thing within the genome of an organism. And so we'll spend a lot of time talking about, where and how do you find all of the different regulatory elements in the genome, like enhancers, repressors, and things like this. We'll also talk a little bit about figuring out, using technologies to figure out what genes actually physically or functionally interact with other genes, or how do these kind of interaction networks form or change across different types of cells or different conditions or with age. And we'll also talk generally about how the cell determines what pieces of the genome or DNA should be turned on or off in different contexts. Something we'll spend a little bit less time on, but obviously is equally important, is kind of the role of genomics in the medical field. And so we'll talk a little bit about, in the human genetics section of the course, about how, for example, genotype generally is related to phenotypes at the organism level. We'll talk about how genome sequence can change in, for example, the case of cancer or other kinds of diseases or with age. And we'll talk a bit about how genomic data, both kind of genome sequence and functional genomics data in general, can help predict, what drugs certain patients might respond to, or people with a certain genetic variation might be more responsive to, and how genomics can be used for general applications like identifying features or behaviors of pathogens or things like this. So next we'll briefly review the basic structure of a gene. And so I really want to harp on this point that the protein coding regions of the genome actually occupy a very small proportion of the whole genome. And so in terms of the human genome, for example, coding exons only occupy roughly 2% of the entire genome, and the rest of the 90% of the genome is non-coding DNA. And so it's still unclear as of today what exactly is in all of that 90% of the genome. But roughly speaking, we think that around 46% of the human genome is repetitive sequence. And so within those repetitive sequences, there can be functional elements that affect gene regulation and cellular activity in general. Roughly 25% of the genome is actually thought to encode different types of regulatory elements in RNA genes. And so in later lectures in the class, in particular in the epigenomics lecture, we'll talk about how to identify and what do those regulatory elements do. But something that hasn't been really that appreciated until in the last 10 years or so is that a significant portion of the non-coding region of the genome is actually attributable to these regulatory elements. And the remaining 27% or so of the genome is thought to be mainly sequences like UTRs or introns of protein coding genes. So a fairly important question that we'll touch on in a number of places in this course is basically the question of how does one genome really encode such a, in the case of humans anyway, such a huge diversity of cell types, and maintain distinct cell identity and function throughout the lifetime of a human. So, because again, for the most part, most cells in the human body have the same genome, then, how does that genome get used in so many different ways for different types of cells and organs? And so in large part, in the part that, we'll explore in this class, is part of that reason is because of gene regulation, right? And so obviously we know from many of our other courses that there's many different types of, regulatory control of genes and proteins, right? And so the level at which we'll talk at for most of this class is really at the level of transcriptional gene regulation, which basically just refers to this part of the cellular machinery and genome, which deals with the regulation of how genes are transcribed. And so some examples of how that might be done is, for example, the cell using various types of machinery to control access to different promoters at different times or in different contexts, through the use of, and basically through the use of things like activators or repressor proteins, or DNA methylation. And so, of course, there's a lot of other forms of control that we won't really touch on, things that are related to post-transcriptional control, translational or post-translational control as well. And so the underlying theme here is that various types of DNA sequencing technologies can really help give us insight into, these different methods of control through technologies like what's called ribosome footprinting and things like this that we'll also touch on later.
I want to take a few minutes now to review the basics of transcriptional regulation. And so a really important point to make is that in the human genome, as well as most complex genomes, only about 2% of the genome actually codes for protein coding exons. And so obviously what that means is that 98% of the genome is non-coding. And so a big open question that we haven't been able to answer completely to date is the question of what does the rest of the 98% of the genome do? And so what we've been able to decipher so far is that about 10 to 20% of the human genome is regulatory. And so that means that it encodes roughly somewhere on the order of 1 million regulatory elements. Those regulatory elements include things like promoters, enhancers, insulators, non-coding genes, and other types of elements. And so those elements are obviously involved in things like precise control of gene expression, as well as in helping dictate which genes should be turned on and off to then further define and distinguish the different types of cells in the human body. And so as I mentioned on the previous slide, the major role of a lot of those non-coding elements in the human genome is to basically facilitate transcriptional regulation. So again, just to briefly review the process of transcription, typically transcription of genes start with some set of chromatin regulators that basically come in, and in the case of activating transcription, they typically open up chromatin and basically move, for example, histones around to increase the accessibility of, for example, the promoter and transcription start site, such that they can be accessed by the transcriptional machinery. What then typically happens is that some transcription factors come in to bind to DNA binding sites that are either at, for example, distal enhancers or at proximal promoters. Transcription factors typically bind to open or accessible DNA sequence. And so after transcription factors bind and, for example, help recruit the basal transcriptional machinery, you basically get a complex of transcription factors forming, which then eventually recruit RNA polymerase and enable transcription to happen.
And so, this is going to be a really frequently recurring theme in the class. Again, transcription factors, at least DNA binding transcription factors, are proteins that bind either specific or not so specific DNA sequences there and basically affect transcription in either a positive or negative way. Depending on how you count it, or whether you define, or whether you're counting DNA binding domains or whole transcription factors, there's roughly somewhere on the order of 3,000 transcription factors in the human genome out of the 20 to 25,000 protein coding genes. And it's worth pointing out that, number one, only a subset of transcription factors may be expressed and floating around in the cell at any given point in time. And transcription factors, more importantly, are oftentimes combinatorial in their nature. And so what that means is that if you have two transcription factors present in a cell, they collectively might do something different than, they might behave differently than if they were expressed in isolation. And so basically they could work, for example, synergistically or antagonistically to yield kind of combinatorially different types of outcomes in terms of gene expression. And so it's worth pointing out that a lot of people have spent a lot of time trying to figure out for each given DNA binding domain of a transcription factor, what is the sequence that that particular DNA binding domain binds to. And so it varies quite a bit in terms of, the particular family of transcription factors that you're looking at. But by and large, most DNA binding domains, individual DNA binding domains, generally speaking, recognize sequences on the order of 4 to 6 is probably the most common. But depending on whether you're talking about DNA binding domains that form dimers and so on, that kind of recognition sequence can run up to like 20 base pairs. And so these transcription factor binding site preferences are typically represented as what's called a sequence logo. So that's basically the big logo of ACs, Gs, and Ts that you see in the bottom right-hand corner of the slide. And so basically the idea is that the x-axis kind of represents the position of a given base in the recognition sequence, and the height of the letter is kind of proportional to what's called the information content, or in some sense the relative frequency of how often you see a letter at that given position in the binding sequence. And so the main point here, and one of the main reasons why understanding DNA binding transcription factor behaviors is so difficult, is because these sequences are super short. And so this sequence you see here, for example, is only 8 base pairs long. And so if you think about it, if a given DNA binding domain has a sequence preference that's really only described by some 8 base pair sequence logo or motif, then the chances of seeing that 8 base pair sequence many times across the genome, even in places where you might not think that transcription factor should bind, is actually really high. And so a huge problem in regulatory genomics is really understanding, kind of distinguishing, okay, even for a given DNA binding transcription factor, even though it could bind to millions of places across the genome, in practice, through some of the technologies that we'll talk about later in this class, a given transcription factor might bind only on the order of like hundreds of those positions, or in the case of like master regulators, it would be more. But generally speaking, we think that transcription factors bind to a lot fewer locations in vivo than you might expect just based on looking at the sequence preferences of these TFs. And so a big question is, why is that? And so I briefly talked about promoters and enhancers. So here I'll kind of more clearly define what exactly they are and where they are. And so promoters are, generally speaking, the so-called upstream region of DNA in terms of the transcription start site. And so promoters are typically found, 100 to 1,000 base pairs upstream of the TSS. And generally speaking, encodes a lot of binding sites or a lot of sequences that are recognized by factors that are associated with RNA polymerase. And sometimes they contain sites that are more responsible for like tissue-specific or cell type-specific binding sites for transcription factors, but mostly it's promoters are, more or less like general purpose and are involved generally in recruiting kind of less cell type specific or context specific regulatory machinery that helps you start transcription. And so generally speaking, there are, as many promoters, at least, as there are transcription start sites in the human genome, but many genes have more than one TSSs. And so in contrast, enhancers are also regions that are, they can range in size, but they're typically 100 to 1 kb segments. It's worth noting that some enhancers, there's so-called super enhancers can stretch like tens of kilobases long. And so these, the size of these elements is still relatively small, but the main difference with promoters is that enhancers can really sit far away from the gene or from the transcription start site that they interact with. And so people have found enhancers that act over like a megabase away on the genome, which is really far. Kind of more importantly, enhancers can be found actually upstream or downstream of the transcription start site. And oftentimes they're kind of direction invariant. And so what that means is that if you take like an enhancer and you like flip it in direction, oftentimes it'll still work almost as well, or basically indistinguishably from the original orientation. And so these enhancers are typically, they typically contain a lot of individual binding sites, again, of DNA binding transcription factors. And enhancers generally act to increase or enhance gene expression of their target promoters. And so obviously one question is, well, how do these enhancers operate at such long distances? And so an example of a very simple mechanism it does so is through looping. And so here in the diagram on the bottom right, you can see that even though the enhancer represented by the E box can be very far away from the promoter, the P, through looping, basically an enhancer can be brought into close proximity of the promoter and basically help recruit transcriptional machinery to that particular gene. And so here's just a kind of a classic example of a promoter and kind of regulatory elements that you might find close to the transcription start site. And so +1 in this on the x-axis basically represents the TSS. And you can see that there's kind of, there can be multiple regulatory elements both immediately upstream and downstream of the TSS. And in contrast, here's an example of what an enhancer might look like. And so it also, again, is kind of rich in transcription factor binding sites. It's also worth pointing out here that transcription factor binding sites don't, they can actually overlap. And so you can have binding sites of multiple TFs in close proximity overlapping. And what that functionally means is that, why that happens is that it might occur in the case where multiple transcription factors need to synergistically interact in order to enhance gene expression. What can also happen is that you can have transcription factor binding sites of competing, or you can have DNA sequences corresponding to TFs that compete with each other. And so, if one TF comes in and binds to its binding site, it might actually occlude and prevent another TF from binding to that site. And so that kind of competitive behavior can lead to kind of some interesting transcriptional dynamics depending on how, depending on what the concentration of these TFs is in the cell at a given time. And so, for example, in mouse, people have found enhancers where if you delete them, they lead to substantial reduction in the size of a mouse, for example. And so it's pretty remarkable that even single base pair substitutions can have very large-scale phenotypic effects on an organism. So here's an example of just like literally a single base pair substitution that can lead to malformations of different limbs in an organism. And this again is, probably corresponds to a single base pair change of one transcription factor binding site for one TF. And so these, it's really hard in general to be able to predict the effect of single point mutations or changes in the genome. And so that's why, the study of regulatory genomics is really so difficult. let's briefly discuss the structure and function of repetitive sequences in the genome. And so we spend a lot of time in this lecture talking about individual transcripts, the regulation, what type of regulatory elements control them, and so on and so on. But we're actually ignoring most of the genome. And so, again, protein coding exons take up about 2% of the genome. Non-coding RNAs, 2%. Regulatory elements, somewhere 10 to 15%. And so that leaves a large part of the genome that is either devoid of function or at least devoid of function that we understand at this current point in time. And so what's really interesting about the human genome and many other genomes as well is that a large part of the human genome in many ways is composed of a repetitive sequence, or what we've identified as a repetitive sequence. So I believe in the human genome, for example, approximately 50%, so that's 50% of the genome is roughly repetitive in sequence. And so what you're looking at here, and you'll see this kind of diagram in multiple lectures later, this is what's called a UCSC genome browser track. And so the UCSC genome browser is a very frequently used tool in genomics for visualizing genome sequences as well as their annotations of different regulatory elements. And so here, generally speaking, the x-axis represents one portion of the genome sequence, and different rows of this diagram correspond to different annotations. And so here on the top part of this diagram, you see basically annotations of different genes and their gene names. And in the lower half of this diagram, you see annotations of different kinds of regulatory elements. And so if you look at the bottom part of the diagram, more specifically, those are basically annotations of where different types of repetitive elements are, at least that were identified by this software called Repeat Masker. And so these signs, lines, LTRs, and things like this all refer to different types of repetitive sequence. And basically the point of showing you this diagram is that you can see that across this section of the human genome that I've displayed, you see a lot of black, rectangles in those rows corresponding to repetitive sequence, much more than, the portion of the genome that's covered by the genes or the DNA hypersensitive sites and things like this. And so again, cumulatively, these kind of repetitive sequences cover like half of the genome. And so it's just worth pointing out that they come in, there's two major different types of repetitive sequences that people often talk about. And so one type is called the simple repeats. And so these are things that look obviously repetitive in sequence. So these are things like AT, 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 or so on, or like, trinucleotide repeats or things like this. And so those type of simple repeats are oftentimes highly polymorphic. And so what that means is that, for these simple repeats where you have like three bases that are repeated like 50 times over, like ATG, ATT, ATG, or something like this, between different individuals in the human population, we can vary widely in terms of the number of times that trinucleotide repeat is actually repeated in our individual genomes. And oftentimes a lot of diseases are associated with, great, large scale expansion of certain types of repeats or things like this. And the other major class of repetitive sequence are what's called the interspersed repeats. And so these are repetitive sequences that actually move around in the genome. So these involve things like transposons or retrotransposons that literally can literally encode certain types of elements that allow themselves to be copied by the cellular machinery and then basically transposed or moved around the genome, or either in a cut and paste fashion or in a copy and paste fashion. And so really in summary, again, depending on how you count it, code the exon regions of the genome occupy a relatively little part of the genome. The regulatory elements occupy a larger fraction, somewhere on the order of 10 to 15%. Repetitive sequences are very prevalent in the human genome as well as other genomes. And oftentimes these types of, these categorizations of different genome sequence elements are very rough in the sense that they're not mutually exclusive. And so a lot of repetitive sequences can actually act as gene regulatory elements and so on.